So, hello and welcome everyone to the week 11 live class session on the course Experimental Biochemistry, which is being taught by Professor Shagati Dasgupta and Professor Shomodaya of IIT Kharagpur, myself Srijan Sen, and today I will be taking your week 11 live session. So I'm sharing my slides with you all, kindly confirm. And firstly, as I always do in all of my classes, we would be starting with a very brief lecture where we would be discussing mostly the important points that are required for solving the assignment and the questions that you need to answer in the upcoming exam as well as the competitive exams that are usually held. So these are helpful in the near future in any exam that you actually take. Okay, so let us start. I guess my screen is visible to you all. Okay, so in today's class we would be mostly discussing about the topic known as protein-ligand interactions. So till now we had been discussing mostly about proteins, the characteristics of proteins, the different types of proteins that exist, the purification strategies. Now coming to a very interesting topic where we have to discuss about the protein-ligand interactions. When we speak of protein-ligand interactions, it was at the 25th anniversary of the Institute of Molecular Biology, University of Oregon. Linus Pauling said that the secret of life is molecular recognition, the ability of one molecule to recognize another through weak binding interactions. So from there on, the concept of protein-ligand interactions began, where binding is the first step that is necessary for any biological response, be it your receptor small molecule interaction, your protein-ligand interactions. Ligands might be your another protein, some small molecules, sometimes peptides, or even macromolecules. So before a macromolecule can perform a function, they normally must interact with a specific ligand. In some cases, like you have myoglobin, binding and subsequent release of the ligand might be the sole function of such a macromolecule. Now to understand the binding, we must consider equilibrium that is involved and how the binding is affected by the ligand and macromolecule concentration, and how to experimentally analyze and interpret the binding data and binding curves. Now, for any kind of binding, we need to understand that there is an equilibrium between the bound complex and the individual molecules. So, the reaction or the complex formation has to be a reversible binding. So, specificity of ligands and binding sites. Ligand binding is often coupled to conformational changes, which you can call as the induced fit model. Also, you have the cooperativity concept, wherein multi-subunit proteins conformational changes on subunit can affect the others, like you see for hemoglobin, and these interactions can be regulated. So what is the interaction with other molecules? Like reversible transient process of chemical equilibrium, you have A plus B in equilibrium with AB. So a molecule that binds to a protein is called a ligand, that is typically a small molecule, and the region of the protein where the ligand binds is known as the binding site. Now, ligands bind via non-covalent forces that dictate the protein structure that allows the interaction to occur, and that is said to be transient. So, binding is a quantitative description, like you can see a process where the ligand binds reversibly to a site of a protein, to the binding site of a protein. You have P and L, and this is the association, that is the formation of the complex. So it is directed by rate constant, associated rate constant, and The reverse process is the dissociation rate constant. So the kinetics of such a process can be described by the association constant and the dissociation constant Ka and Kd respectively. Now what happens after a certain point of time is that the process will reach equilibrium where the association and dissociation rates are equal, and we can write it as Ka into the protein concentration and ligand concentration, which is equal to the Kd into your protein ligand complex concentration. So at equilibrium, this is the case. Apart from that, the equilibrium composition is characterized by the equilibrium constant, that is Ka, the capital Ka, which is also equal to Ka small k by Kd, which you can see over here, and that is equal to your complex formation divided by your protein and ligand concentrations. So when you analyze in terms of the bound fraction, what happens is that the fraction of occupied binding site is indicated by theta. So we often determine this by the binding site, the fraction by theta. So theta equals to the complex concentration divided by the protein ligand complex concentration plus the protein concentration, because the concentration of the protein is known to us and we know the amount of protein that is bound to the ligand. So when we substitute the complex concentration by the value that we got previously, that is capital Ka association constant into ligand concentration into protein concentration, what we get is theta equals to Ka into ligand concentration into protein concentration divided by Ka into ligand concentration plus protein concentration plus the protein concentration. Now if we are eliminating P, we take common and we eliminate it and rearrange. So we get theta equals to ligand concentration divided by ligand concentration plus 1 by Ka. Now 1 by Ka is ultimately the reverse of the association constant, that is equal to your dissociation constant. So you can write theta equals to ligand concentration divided by ligand concentration plus Kd. So in terms of the more commonly used equilibrium dissociation constant, we can express the fraction of occupied binding sites, where the dependent variable, where we know the concentration of the protein. Also, if we know the concentration of the ligand, it is very easy to calculate Kd. So the graphical analysis speaks like the fractions of the bound fraction depends on the free ligand concentration and Kd value. So experimentally the ligand concentration is not, Kd can be determined graphically or via the equation that we just saw. So theta is equal to your ligand concentration divided by ligand concentration plus Kd value, and your ligand concentration is equal to your, more or less closely equal to your total ligand concentration, because here we are imagining that very less number of ligands are binding to the protein, or very less concentration of ligand is utilized in the binding process. So you can understand the binding from the graphical analysis. Apart from that, what other graphical analysis it says is that you can see this equation that is in equilibrium. Now Kd equals to protein into ligand concentration divided by the complex. So now when you arrange it, you put all the values over here and you try to summarize the or shorten up the equation. What you get is complex concentration divided by ligand concentration equals to protein concentration, that is the total protein concentration minus the ligand concentration, the complex concentration divided by Kd. So from there you can find the Kd value from the slope. So y equals to minus 1 by Kd into x plus This is the intercept, so from the slope you can get the Kd value. All that we need to find is the Kd, that is the dissociation constant. So lower dissociation constant indicates tighter binding, higher dissociation constant indicates loose binding. So you can see over here, lower Kd means higher affinity of protein for ligand, and higher Kd means lower affinity for protein or ligand. So the thermodynamic of this binding study says that the interaction strength can be expressed as association constant Ka, dissociation constant Kd. You have the free energy, that is delta G naught, So these are the equations that we already know, that is delta G naught equals to delta H naught minus T delta S. So association constant is equal to your complex concentration divided by the protein and the ligand concentration, and Kd is vice versa. So the another equation is delta G naught equals to RTLN minus RTLN or RTLN Kd or minus RTLN Ka. So what you call a stronger binding is Kd less than 10 nanomolar, and a weaker binding is Kd greater than 10 nanomolar, micromolar. So if you have a Kd value in millimolar range, that is a weak binding, and if you have a Kd value in say picomolar or nanomolar range, that is a tighter binding. So here you can see the different dissociation constants of different protein ligand interactions, like you have avidin, biotin, you have insulin receptor for insulin, then you have the anti-HIV immunoglobulin for GP41, you have nickel binding protein for nickel ions, and finally calmodulin with the calcium ions. So you see, among the highest affinity you have biotin avidin interaction. In moderate affinity you have antibody antigen sequence specific DNA. You have the receptor ligand interactions, and the enzyme substrate falls in the low affinity. Now, when we speak of the Gibbs free energy of binding, so the binding constant is connected to the Gibbs free energy of binding using the equation delta G equals to RTL in KD. So as I was mentioning, KD, that is lower KD value. Yeah, good evening. Yeah, so if you can see that a lower KD value means higher affinity for protein for the particular ligand, and a higher KD value indicates lower affinity. So you can see that delta G naught equals to RTL in minus RTL in Ka or RTL in Kd. So what it says that you have a receptor molecule or a protein and a ligand, and the forward is the association constant, the reverse is the dissociation constant. So Ka equals to 1 by Kd, that is equals to your complex concentration divided by the protein and the ligand concentration. So your enthalpy and entropy would be delta G equals to minus RT ln Ka, which is equal to delta H minus T del S. So minus delta T G by T equals to minus delta H by T plus delta S. So there are two important types of protein ligand interactions where you have the hydrogen bonds, the typical, untypical, charge assisted. You have the ionic interactions, that is the effect of pKa values, the local pH of the binding environment. You have the hydrophobic interactions that are mainly caused by the displacement, or more correctly put, the liberation of water molecules from the lipophilic environment of the binding pocket, and the cation pi interactions. So this is about the idea about the binding, the protein ligand binding. Now, how do we know that a ligand is binding to the protein or vice versa? So for that we have isothermal titration calorimetry. So you can see over here, this is the apparatus for ITC. So isothermal titration calorimetry is a very direct method that measures the heat change associated with the complex formation at a constant temperature. So you see, this is your protein, this is the ligand, or this is the ligand, you have a protein over here, you inject it, you are capturing the change, the heat change that is associated with the complex formation at a constant temperature. So you, from here you can get the complete thermodynamic profile of the molecular interactions in a single experiment, which includes your enthalpy, entropy, Gibbs free energy, binding affinity, and the stoichiometry. So what is the instrumentation? You have a reference cell over here, so the delta T, so it's a constant power to reference cell heater, and this is the power supplied to the sample cell feedback to keep the delta T 0. So this is an adiabatic jacket. You have a syringe over here, you have the sample, and here you're injecting the protein to the ligand or the ligand to the protein. Now you see that this is an exothermic heat change, and you have the protein, you have the ligand, it binds. This is an endothermic heat change, and this is an exothermic heat change. So depending on that, you can actually find out the delta H, the heat change, and from there you can calculate all the other parameters associated with enthalpy and entropy. Okay. So you see, you have a sensor over here, you have a lead screw, you have the injector, plunger, the stirring system. The syringe is placed over here. This is the sample cell, this is the reference cell that will measure the temperature. You have the inner adiabatic jacket. So the review of the free energies, enthalpies and entropies of binding says that delta G binding equals to RTL and KD. But these are the parameters at a particular temperature. Now, if you see over here that it's a logarithmic relationship between the free energy and the binding constant. So let us recall that delta G binding is relative to the standard conditions. And the convenient rule of thumbs is that tenfold change in binding constant corresponds to 1.4 kilocalorie per mole. So if there is a change in the tenfold binding constant, it will correspond to 1.4 kilocalorie per mole. So if there is a change in 100-fold, how much does it change? So mostly you would tell that it is into 10, 14 kilocalorie per mole, but that is not correct because it's a logarithmic relationship. So log of 100, that is 2, so it will increase by 2 times, so that is why it is minus 2.8 kilocalorie per mole. Also, if we need to see that free energy has enthalpy and entropy components, we can calculate them by delta G equals to delta H minus T del S, and from there you can find all the given parameters. So when is an interaction strong? So delta G naught must be large and negative, delta H must be large and negative, delta S must be large and positive, that it should gain more entropy, and delta H should gain new bonds. Only then the interaction is said to be a strong interactions. Okay. Another very interesting technique is the surface plasmon resonance, or the SPR. So SPR, so plasmon, surface plasmon is a longitudinal charge density wave along the surface of two media, where one is the metal, other is a dielectric. So what happens over here is that you have a metal film of a particular thickness. Here you might have a liquid or an air, and this is a plane polarized light. Here this is immobilized with your particular ligand, and you pass the protein. So depending on the binding, it will change the angle. So the angle will change, the angle of the plane polarized light will change, and that is captured by the 2D detector. So if you see the SPR principle, what is happening? So SPR is excited by a plane polarized light, that is totally internally reflected light at the glass or metal surface film interface and the surface plasmon, enhancing the evanescent field amplitude, that is E. Now, so in this particular system, you have a sensor chip, that is the interface takes the form of an exchangeable gold coated glass slide. So SPR is observed as there is a dip in the reflected light intensity at a specific angle of reflection. So when there is a deflection, you get a peak like this, and from there you can understand. So you have a linear relationship between the resonance energy and the mass concentration of biologically relevant molecules. So you have a sensor chip over here. This is immobilized with an antibody. You have passed the antigens. You have the plane polarized light. That is the light source that is passed, and you can catch the reflected light over here. So as the angle changes, so you can see the change in the angle, and this you can draw it in this manner. So this is the association. So when it is getting associated, it is getting, so this is the range of association, and when it starts dissociating, it sharply decreases. So sharper the decrease, lower is the binding affinity, and so steeper the decrease. If this decrease is steeper, it means it's low affinity. If the is not that steep, it is high affinity. So there are several applications of SPR. The physical applications means it measures the dielectric properties, adsorption processes, surface degradation or hydration of polymer films, organic monolayers or bilayers. And several biological applications includes its role in biosensors, for specific biological interactions including adsorption and desorption kinetics, antigen antibody binding, and you have the epitope mapping for the determination of biomolecular structure and interaction of proteins, nucleic acids, viruses, lipid bilayers, tissue engineering, non-specific biomolecular interactions, and biocompatibility. So SPR has several advantages, which includes its ability to perform real-time measurement, where you can understand the association as well as the dissociation. Now, insight to dynamic nature of binding system and layer formation can be understood from over here. Use of selective slides to study the binding event. It eliminates the need of for labeled reactants, and it has exceptional sensitivity. So small quantities of purified reagents are basically required. Also, this method has a few disadvantages, which includes its lack of sensitivity when monitoring low molecular weight adsorbents. Weight limiting factor is an issue for mass transport affecting the kinetics analysis. And in order to improve these disadvantages, it is usually coupled with the atomic force microscopy and mass spectrometry. Okay. So this is all about your surface plasmon resonance. So two most interesting experiments we have studied. We have another two interesting experiment. One is your electrophoretic mobility shift assay, and the other one is anisotropy, fluorescence anisotropy. Okay, now getting to electrophoretic mobility shift assay, it is also known as the gel shift assay or the gel mobility shift assay. So it is a technique to detect whether the protein is attached to a DNA or RNA or nothing, or not to anything. So as we know that there are many cellular processes where the protein needs to interact with the DNA and RNA, like we have the uh, say transcription factors that regulates the transcriptional machinery and all. So originally utilized broadly in the investigation of sequence-specific DNA binding proteins such as the transcription factors, as I was just mentioning. You have EMSA has been additionally developed to explore DNA protein interactions, RNA protein interactions. And it is a very sensitive method using radioisotopes to label nucleic acids and autoradiography. It is possible to use very low concentrations and small sample volumes. So how do we go with it? So the purified protein and the cell crude extract is usually incubated with the isotope 32P isotope level DNA or RNA probe, and the complex and the unbound probe are isolated on the denaturing polypropylene gel electrophoresis. So a mobility shift assay is electrophoretic separation of protein DNA or protein RNA mixture on a polyacrylamide or agarose gel for a short period, and the speed at which different molecules move through the gel is determined by their size, charge, shape, and the complex formation. So how do we do the transformation, the titration? So as we know that we are going to have a DNA that is interacting with a protein. And for that to happen, we need to have enough DNA and protein in the solution. So if you see that this is a gel that we have run. Now, the lower lane is basically the free DNA. So where no protein is bound. Once the protein DNA complex is formed, you see there is a shift. The bare DNA runs over here, and when on increasing the molecular weight, the protein starts running at a higher level. And when the DNA is bound to two proteins, it is possible for the DNA to bind to two proteins. So you see another shift over here. And you call this shift of the DNA protein complex and the DNA bound to another protein is known as the super shift, the lower lane being the bare DNA. So the DNA is labeled. You cannot see any free protein over here. Why? Because the protein is not labeled, rather the DNA is labeled, so you can see only when a protein goes and binds to the DNA, and you can see the shift in the bands. So the applications of IMSA includes that it is used to study the conformational changes in DNA. For example, a IMSA 2D variant with electron microscopy is used for the characterization of conformation. You can analyze the cellular extracts for the presence of certain DNA binding proteins, and bound and free DNA are separated using IMSA. And it proves useful for stoichiometric analysis, number of protein that binds per the DNA fragment. Also, it is used to study whether the transcription factor is attached to the DNA or not, or if the transcription factor is attached, it is our targeted and not, and their activity. So the advantages of IMSA is that it is used with a wide range of nucleic acid sizes, structure, as well as wide range of proteins. It is a simple method to perform and is used for qualitative analysis. It proves useful in the detection of protein that binds to DNA and cause difficulty in migration or fragment on gel. It is a highly sensitive method and usually low cost, and no special equipment is required, and it is also possible to use both crude protein extracts and the recombinant proteins. The drawbacks of IMSA are basically it's not an appropriate method for the kinetic studies. You cannot understand the kinetics from here. Dissociation is one of the major drawbacks of IMSA, so you need to run the gel it's a very sensitive experiment, first of all, and any kind of contamination in RNA or DNA can just mislead you to a different result. So that is one major point. Also dissociation, because you cannot keep the gel in standby mode for a long time, otherwise it will dissociate and it will give you a misleading or an inappropriate result. So the last technique is basically the fluorescence polarization, the fluorescence anisotropic experiment. So what is polarization in a fluorescence experiment? If we see that polarization is equal to your intensity in the parallel direction minus the intensity in the perpendicular direction divided by the sum of the two intensities. And your anisotropy, or R, is represented by the total the difference between the parallel intensity and the perpendicular intensity divided by the total intensity. You can also write it as the total intensity can be written as the parallel intensity plus the perpendicular intensity into 2. So 2 into perpendicular intensity. So the difference between the two intensities divided by the intensity in the parallel direction plus twice the intensity in the perpendicular direction. So anisotropy is preferred because it contains the total intensity, and that can ease up the entire process. So keeping this equation in mind, what is the total intensity equal to? It's a question, why is the total intensity equal to this value? So for the reason of symmetry, the component polarized in the x direction and the y direction have the same intensity, that is the perpendicular intensity. But in a given geometry, we can detect only one polarized in the x direction. So parallel is in the, you can see if this in direction, this is an excitation, so you can catch it in the z direction. So for that you have parallel, and the perpendicular ones would be x and y, but you can catch the intensity only once, so it is twice into that intensity. Now, anisotropy is related to molecular motion. So far we have considered frozen molecules. However, in reality, the molecules are mobile and they are entropically, they have high entropy. And the rotation changes the orientation of the dipole theta. So changes in theta are reducing the anisotropy caused by photoselection. So if you look into this equation where rho is The rotational correlation time divides rotational relaxation time, which is the time for a given orientation to rotate through the angle. So for a spherical molecule, you have rho equals to viscosity into volume divided by r into t, so it's velocity, so which is equals to 1 by 6d, that is the translational diffusion coefficient. So viscosity into v divided by rt. And for a globular protein, you have rho equals to viscosity into mass multiplied by the addition of partial specific volume plus degree of hydration divided by RT. So the measurement of fluorescence anisotropy is that this is a vertical excitation that you are giving, and this is the detection zone. So the plane defined by the direction of excitement and detection is called horizontal. Now, the measured intensities are identified by the two indices describing the orientations of the excitation polarizer and the analyzer. So we have to account for the different sensitivities of detection of individual polarizations. So if you see that anisotropy is equal to intensity in the vertical direction, vertical direction plus twice into, so this is the total intensity in the vertical direction plus twice into the intensity of the vertical horizontal direction, where you have the Swedberg's constant in the vertical by the horizontal direction. And this is the equation of anisotropy, which you can use to plot these particular graphs where you are plotting the photon counts per channel, and here you are plotting. When you put all of these values over here, you get a curve like this, anisotropic curve, and that is anisotropy versus the channel. So the scattered light can cause higher anisotropy because it is completely polarized. So it is necessary to minimize scattering for accurate anisotropic measurements. So polarization methods are suited for the study of aggregation states of proteins, like in the case of protein dimer monomer equilibrium. So either intrinsic protein fluorescence, or by labeling the protein with a suitable probe, one would expect the polarization of the system to decrease upon dissociation of the dimer into monomers, since the smaller monomers will rotate more rapidly than the dimers due to the excited state lifetime. Hence, for a given probe time, the polarization of the monomer will be less than that of an isomer, which will be also less than for a polymer. So these are the four important techniques that is associated with the protein ligand binding. First, we started with firstly we started with ITC, or isothermal titration calorimetry, from where we can get all the parameters regarding the thermodynamic parameters, like we have the entropy, enthalpy, Gibbs free energy, the dissociation constant, the association constant, etc. Then we moved on to SPR, from where we can get an idea about the kinetics, followed by IMSA, where we can understand also the stoichiometry of the proteins that is binding to the DNA. And finally this fluorescence anisotropy, which will also give an idea about the protein ligand binding. So now, do you have any question till now? Any questions on this particular techniques? Yeah, it's okay, but this lecture is being recorded, so you can also check back on YouTube. Okay, so this will be uploaded in YouTube. You can check there. So the good thing about this is that these lectures get recorded even if people miss classes or they are late, they can definitely go and check them in later on. So this is okay. On which channel? I think the NPTEL team shares with you. But anyways, I am so if you write by Sri Johnson, you can get it. Otherwise, I don't even remember my channel's name. I don't remember it usually. Wait, I am mentioning it. Just give me a minute. Yeah. So it is bio. So this is my channel name. You can go and check all the lectures over there. It is it is there only, so I guess you can find it. Won't be much of a problem. So in each of my class, I usually what I do, I first talk about the lecture and then I discuss the questions. So that questions are discussed in a manner where I put forward the questions and you give me an answer, and then we can discuss it if you people are making mistakes. Otherwise, it is good. So I would be starting with the questions very shortly. So it's an issue, not a big issue. Okay, so whenever I'm giving you the questions, you answer me in the chat box, whichever option you feel is correct. And if you're not being able to understand it, I will surely explain it. Okay, so give me just 2 minutes. I will start with the questions. Okay, so let us start. You can answer me in the chat box. So let us start with the questions. Yeah. So the first question. What is the answer to the first question? Where you have been asked the units of K on and K off. B. Anyone else who wants to give an answer? You say it's A. So molar, second inverse, molar inverse, second inverse, and second inverse. And you have molar inverse, second and second, and second inverse, and molar inverse, second inverse. So for K off, we usually know it is what? So KD equals to K on by K off, right? So if that is the case, we write KD in molar inverse. So the answer has to be in molar inverse. So second inverse gets cancelled and molar inverse remains. So option B is the correct answer. You have to remember KD equals to K on by K off. Okay, next one. Which of the following technique is used for studying DNA protein interaction? Which setup does it require to perform the experiment? So tell me. Which experiments can you perform for protein DNA interaction? SPR, UV visible spectroscopy, IMSA native polyacrylamide gel, IMSA SDS page, DNA agarose gel electrophoresis, and urea polyacrylamide. So if you have studied about any other answer, I got one answer. No. So if you see the gel that we usually run for IMSA is an acrylamide native that does not contain SDS in it. So it's a normal polyacrylamide gel or a native polyacrylamide gel. We have already studied that IMSA can be used for studying it. You cannot use an UV visible spectroscopy for that. You have to run an IMSA. So B or C is mostly correct. And you have SDS that you definitely cannot run for nucleic acid. So option B is more correct as an option. Let's go to the next question. Now tell me, which of the following experiments can be used to determine the binding kinetics under favorable conditions? EMSA, ITC, fluorescence anisotropy, surface plasmon resonance. The binding kinetics, K on, K off, KD. Yes. Very good. So all the options are correct. Yes, you can also use fluorescence anisotropy to get the KD value. Why not? So all the options are basically correct. So all of these techniques can be used to determine the binding kinetics at different conditions probably, but not at the same conditions. But yes. Anyways, so let's see. Let's go to the next one. Enthalpy of a binding reaction can be determined from. Okay, I got one answer. Yes, enthalpy can be determined from isothermal titration calorimetry. Fantastic. Right. Let's go to the next one. The dissociation rate constant K of for a DNA protein interaction is determined by EMSA. The intensity of the free ligand can fit into which of the following equations to obtain K of? Let's see if you can do this. Otherwise, this is also a part of, this is another equation. So just write it down somewhere if you are not aware of it when I will tell the answer. Anyone else with a different answer? So option A is the correct answer. Intensity equals to I naught into 1 minus e to the power minus k of into t. Okay. So what is the total heat that can be expressed as? So from this graph, you have to calculate the total heat. So how are you going to calculate the total heat? That was quick an answer. That is good. Any other answer? No? Yes. Option C is the correct answer. The total of the hits will be this one, the addition. The next question, which of the following statement about protein ligand binding is true? Which of the above statements about the protein ligand binding is true? Let's see what are the options. K is independent of such conditions as salt concentration and pH. When all the binding sites are occupied, the Ka equals to ligand concentration. Ka is increased, the binding will be tighter. The larger Ka means weaker affinity. Okay, got one answer. Larger. Okay, let's start from the first. Ka is independent of salt concentration and pH. That is incorrect. When all the binding sites are occupied, the Ka equals to ligand concentration. That is also incorrect. If Ka is increased, then the binding will be tighter. Yes, that is correct. If Ka increases, tighter binding. If Kd increases, loose binding. Similarly, if Kd decreases, the binding is tighter. Kd decreasing means Ka increasing, and larger Ka means higher affinity, not weaker affinity. So option C is correct. Now, question 8. A cytochrome P40 enzyme binds with a newly synthesized drug molecule with association constant 10 to the power 7 molar inverse. What is most likely Kd in the presence of a competing inhibitor? Keep this thing in mind, in the presence of a competing inhibitor. D. Okay, let us see now. So Ka is given as 10 to the power 7 molar inverse. So Kd in the normal scenario would have been 10 to the power minus 7 molar. That is absolutely correct. The approach is absolutely correct. But what will happen in the presence of a competing inhibitor? In the absence of a competing inhibitor, you do not have to worry much, because it is simple, the inverse of the Ka value. Okay. But when there is a competing inhibitor, what happens? For one particular site, there are two molecules that is competing. So the affinity ultimately decreases. So over here what we see is that there is only one value that has higher Kd than the others. So 10 to the power minus 6 molar is a correct option when in the presence of a competing inhibitor. If it is in the absence of any inhibitor, it is 10 to the power minus 7. For a non-competitive inhibitor, it is also even 10 to the power minus 7. But when it's a competitive inhibitor, the Kd value increases. The next one, this is a match the following, should be a simple one. Tell me. No answer. So let us see. Isothermal titration calorimetry. So ITC resonant. Okay, let's start from the description. It is a resonant oscillation of conduction electrons between negative and positive permittivity in a particle of incident light. So 3 is 1, surface plasmon resonance. So 3, 1. Okay, so 1, 2. So 2 and 3. Okay, so 3 is 1. The next one, it's a physical technique used to determine the thermodynamic parameters in a solution. Okay, it is used to detect protein complexes with nucleic acids. This is EMSA. So 4, 3. So 3, 1, 4, 3. So option D is the correct one. So one, the resonant oscillation is fluorescence. Is your surface plasmon resonance. The technique used to determine the thermodynamic parameters is ITC. Detect protein complexes with nucleic acid is EMSA, and the phenomenon where light emitted by a fluorophore is fluorescence anisotropy. Let's go to the next one. If the free energy delta G for the binding interaction is known, we can calculate what? So what can you calculate if the free energy is known? Association constant, dissociation constant, K on, K off. Yes. So if the free energy is known to us, We can calculate the association constant, the dissociation constant. The other two cannot be calculated because for that there are other parameters that are involved alongside the dissociation constant. So just from the free energy, it is written only from the free energy. Only from the free energy you can calculate the two constant values. So option C is correct, not option D. Okay, the next one. Now this is interesting. The k on and off values of ligands X and Y is given. So for ligand X you have on value and the off value, and for ligand Y you have the on value and the off value. Now which of the following statement is or are true? X binds E more tightly, Y binds E more tightly, Xe complex is longer lived than Ye complex is longer lived than Xe. So which of the following statement is or are correct? Okay, for this remember, whenever there is a binding, when you have to talk about the binding, that is individual molecules, then look at the K on. When you are talking about the complex, look at the K off. Okay, so X binds E more tightly, Y binds E more tightly. So if you look at the K on value, we will be getting an idea about the binding kinetics. So 10 to the power 7, that is Ka association. So K on is for binding. So Y binds E more tightly. So 2 is correct. And you see that the K of value, so lower the K of value, longer will be the uh, lifespan of that particular complex. So this value is lower than this. So Xe complex is longer lived than Ye. So remember one thing, that for understanding about the binding, look at the K on. So you will be getting how tightly the particular molecule binds to the ligand or the enzyme. And for looking at the complex, from the complex perspective, you have to look at the K off value. The next question. In ITC, for a titration experiment, determine the dissociation constant. It is necessary to vary temperature, the ligand concentration is varied, protein concentration kept constant, protein concentration varied, ligand concentration kept constant. The concentration of free ligand, free protein and bound complex must be determined. Isothermal titration calorimetry. So it has a very simple answer. The ligand concentration is varied and protein concentration kept constant, or the protein concentration is varied and the ligand concentration is kept constant. So 2 and 3 are correct. The next one, a researcher designed a new drug molecule and wanted to study the interaction with the human peptidase enzyme. If the peptidase drug molecule dissociates with an off rate of K of 1549 second inverse, then what is the time constant for the reaction? So you have to find out tau from the K of value. Okay, so it is 1 by 1549. So 1 by 1/49 is how much? Point, no, that is 0.645 milliseconds. So it is this one. B. So there might be a calculation error. It is 0.645 milliseconds or 645.5 microseconds. So D is correct. Okay, the next one. The dissociation constant for a protein DNA interaction is determined by EMSA. The intensity of the free ligand is given, and you have to find out the K of. So if you can understand this, try using this equation. The equation where I said this one, try using this equation and solve it. I equals to I naught 1 minus e to the power minus k of by t. So try using it and give me an answer. Okay, so if you use that formula to calculate it, the answer would be 0.1 minute inverse. So try doing it. Okay, I said the formula is this one. Yes, this one. So you can use this formula to get the answer. And the last question is this. So the protein concentration is 10 micromolar, and the expected which is more than the expected Kd. So we have to determine the Kd from the ligand concentration and the complex concentration. So how are you going to do it? So if you do not remember the formula, I'm just telling you. Yes. You can use this formula where Kd is just the reverse of that. I think I have the formula for it. Yes. So this is the formula which you have to use. Okay, so this is the formula. So use it and give me an answer. Okay, you have copied it, the formula. Now try solving this. Yeah. Anyways, you can do using that formula. So the answer is so basically the answer is B. So the answer is B. Okay, try doing these two problems by yourself. I'm sure you will get the answer. Okay, so try doing this because there will be a few this type of questions in your exam, so these are good for your practice. Okay, so do you have any questions? Any questions from anyone? So you see what you have to do now. Okay. So like this you have to plot. Okay. So here you have to plot like this, and you have to find the slope. Slope is -1 by kd. Okay. So you have to do it. It's a graphical analysis. So you have to plot this by this, and then you have to draw. You will might get a straight line, and from there you have to find out the slope. Okay. Um, Possibly this question won't be there, but this type, I mean, what do I mean by this type is that you would be given one ligand concentration, probably one uh, complex concentration, and the total protein concentration, and then you have to find out the kd. The graphical analysis won't be there, but yes, the a question regarding this type of equation, using this kind of equation, might be there. Okay. Okay, thank you. We will again meet in the next week.